swigging out of it. Carly, you really let me down. It was our microphone. The only thing is, I have to say, if I get a microphone in my hand, I really am desperate to sing, so I won't stay on here too much longer. I'm just going to keep talking a little bit about Damer while he sorts out his microphone. So Damer's come up from the Royal Veterinary College in London to share some information on parasite vaccines with us. He's going to introduce us generally, first of all, and then focus down on coccidial vaccines. And you guys in bacteriology and virology have no idea about the tough nuts that we've got to crack, particularly with multicellular parasites, but as well with protozoa. So I'll just hand over to Damer. Okay, thanks.
So is this working now? People can hear me? Good? Anyone goes to sleep at the back, I can shout at you then. That's good. Okay, so the brief for this session was veterinary vaccines, specifically those for parasites. I've gone for a bit of a hybrid session, so as Jackie said, we've got a bit where we start off looking at existing vaccines for parasites, and it's sad to say that's not a particularly big catalogue. We'll then go on to focus on some work we're doing, largely within SAPHIR, but actually overlapping with some work in PARAGONE as well, to develop what we hope will be new veterinary vaccines. So I can start off by saying parasites are pretty much everywhere. If we look around, many of us may not know it, but we may well have parasites. And if we look at the definition of parasitism, it's pretty broad. We see it's an intimate relationship between two organisms in which one, that's the parasite, lives at the expense of the other, that's the host. And of course, if we take that definition, you actually have a pretty broad range of organisms that could be parasites. Anything from viruses, some bacteria, we've got some protozoans, helminths, arthropods. You might even say some people, perhaps. I'm going to be a bit more traditional than that. I'm going to focus on what we normally consider to be parasites. So we'll be looking here at protozoans, helminths, and arthropods. But having said that parasites are everywhere, sad to say it's not the case that parasite vaccines are everywhere. There's very few parasite vaccines. Indeed, vaccines that protect against parasites like plasmodium, so that's the cause of malaria, against trypanosomes, against hookworms, ascarids. We don't have vaccines, so not commercial vaccines, for any of these parasites. And it is important to note that parasites remain a serious cause of both morbidity and in some cases mortality. Now this is quite an old table. It's the top half of a table showing the top 20 diseases or pathogens, and that's ranked according to their impact on the poor. And of course, I think the point is, if you've got parasites or viruses or bacteria that impact on livestock, then inevitably they will impact on the poor. What I've done is highlight with red arrows all of those on this list that are parasites. We've snuck one in with the blue arrow. It's not actually a parasite, but it's transmitted by a parasite, so we can hopefully let that one go through. But you see, if we look in West Africa, ectoparasites are a big impact. We've got quite a broad group here, gastrointestinal parasites. There's some specific pathogens here, liver fluke. I mean, we know about liver fluke in this country, of course. There's problems here with liver fluke. Trypanosomes. If we move to Eastern Central Africa, we've got East Coast fever, so looking at Theileria. Again, ectoparasites, gastrointestinal parasites. Hemoncus, a specific example. If we look to South Asia, coccidiosis, and these are not ranked in any order, in alphabetical order, but coccidiosis is obviously one of my personal favourites. Again, we have liver fluke, Toxocara, and again, when you go to Southeast Asia, you see the same candidates appearing as ectoparasites, gastrointestinal parasites, and here Toxocara. So it's fair to say there's lots of parasites out there that have an impact. So why do we not have vaccines that protect against all of these pathogens? I think there's many reasons. For a start, they have quite complex life cycles in many cases. They might have intermediate hosts, reservoir hosts, environmental phases, eggs, cysts that survive, perhaps in the environment or in other hosts. Some of them have options where they can transfer between different hosts, different reservoirs. Some parasites also have complex antigenic profiles, perhaps the ability to change their protein coat or expressing different proteins at different stages of their life cycle. Many of them have evolved ways in which they can escape natural immune responses. So again, it could be, for example, by changing those antigenic profiles. It might be where they reside, where they replicate, how they escape immune killing. And above all, most of them have fairly rapid levels of evolution, certainly more rapid than we do, than livestock does. And so, of course, if we try to evolve to control these pathogens, they can evolve much quicker. And in many cases, there's a lot of pre-existing genetic diversity. But, as we've heard from Camille, and we hear throughout this meeting, there's a strong economic and strong social driver to have vaccines to control parasites. And of course, we're now also very interested in welfare of livestock as well. Now, I'm going to go through a few shining examples that we have of anti-parasite vaccines for use with livestock. But as I say, there's not very many of them. So we do look at the vaccines that are out there. We see examples that are perhaps whole parasite vaccines. So this may be with a live parasite that we deliberately infect, say, livestock with. You allow it to replicate, and that can stimulate a natural type immune response. The examples where we can look at subunit proteins. So these are antigens, but maybe native antigens that we recover from the parasites themselves after we've cultured them, either in animals or perhaps in cell culture. And there's also recombinant antigens. Now, of course, these are very attractive because they're cheap, they're easy to produce, but there's a very small number of them that we actually have that are effective, that have made it to market so far. And that's why in the future we want to have many more vaccines. We want to have vaccines that target pathogens we don't yet have vaccines for. We want to use different approaches that may be cheaper or easier to use, perhaps DNA vaccines, dietary vaccines, things that we can actually feed to animals to vaccinate them. And also vector vaccines. And I put that one in bold because the second half of this talk I'm going to be focusing on how we're trying to develop a new vector vaccine approach. So we look at these examples. Well, talking about whole parasites, in particular whole live parasites. These are parasites that you deliberately infect an animal, they go through their life cycle and they stimulate a natural type immune response. Now, the parasite I'm going to start with is Eimeria. That's the cause of coccidiosis. I think many of you may have heard of Eimeria, but just a bit of background for those who don't. And the reason I'm talking about this is that you'll see Eimeria popping up several times in this talk. So Eimerian is a protozoan. It's not a multicellular organism, as you say, although that is a multicellular stage we have there. You've actually got 8 parasites hiding in that egg. So it's a community, you might say. But this is an oocyst. That's the infectious stage of the parasite. It survives in the environment for a long period of time. And pretty much every chicken that's produced in the world is likely to be exposed to oocysts like that. I mean, how many chickens do we think there are in the world? Anyone have a guess? It's like a game show.
4 billion.
Higher, yeah, so we're going to get to that. You stole my line. Oh. Have we any options? 2 billion. Higher or lower?
50 billion.
Oh, we're getting close now. 50 billion. Higher, lower? Higher. Yeah, it depends where you look, but it's about between 60 and 64 billion chickens in the world. So, I mean, that says if you're a parasite that infects chickens, it's a wonderful world. There's so many potential hosts out there. And, of course, the way we rear chickens, we constantly have batches of young, immunologically naive chickens coming along. It really is fantastic. And, of course, what happens if they're infected, if it's not properly controlled, some animals will become sick, and you may see infection or disease occurring that's affecting perhaps the small or the large intestine, depending on the infecting species. Now, the next slide is a parasite life cycle. Don't be afraid, I always find that every parasitology lecture has at least one life cycle, and this is it. I'm not going to go through it all in detail, but the point is, Eimeria life cycle is multiple stages. So you've got stages that happen in the environment, you've got several stages that happen inside the host, where you've got periods of asexual replication and sexual replication. Now, the reason to show this is if you look to the literature, we can see that that first stage of parasite replication in the host, in this case the chicken, is able to stimulate a protective immune response. Yep, so we can infect with that parasite, you could perhaps control the parasite with drugs, and actually if you challenge later on, there is some immunity there. So that's important knowledge. But actually we also see that later asexual stage also stimulates robust protective immunity. And so do the sexual stages at the end. So of course what that means is if you have this complete life cycle, you've got an opportunity to stimulate a really robust immune response. And of course, this has been used to produce the very first vaccines against Eimeria. So this is one of the first vaccines that are out there, certainly for parasites. The first was introduced in the US. It was introduced in 1952. And it was a forerunner of this vaccine, Coxavac. And it essentially contained oocysts of one parasite species. It was Eimeria tenella. And it was a very crude suspension. So essentially, they purify oocysts of the parasite. You prepare a very dilute dose, and then you give those low oocyst doses to chickens. You allow the parasite to recycle through the birds. They become immune. And ultimately, when they then face the challenge, perhaps from the wild populations, they should be protected. But there are some problems. We can't produce Eimeria in cell culture. The only way you can do it is to produce by infecting chickens. So certainly here in the UK, for example, more than 250,000 chickens are used every year just to produce vaccines for other chickens. So it's a big business. And that means because of this inherent limitation, the way in which we produce the vaccine, it's traditionally been restricted to use with breeding chickens and laying chickens. It's because we keep them for longer, and relatively speaking, they're a bit more valuable to us. Now, it's really interesting. There's a big change happening in terms of Eimeria vaccination at the minute. So if I'd given this lecture 20 years ago, there I am. So if I'd given this lecture 20 years ago, I'd have been given perhaps a similar topic. We'd have had those same sort of wild-type vaccines in the US with lots of drugs. I can see that I'm celebrating. Yay, coccidiosis vaccines. If I'd given the same talk 40 years ago, again, it was perhaps similar. We had less advanced vaccines, but vaccines were still out there, and we primarily relied on drugs. But what's changing in the last couple of years, and this is actually being led from the US, is this drive to remove antibiotics from livestock as we produce them. And it's a bit of a surprise, because historically, I think, correct me if I'm wrong, but historically, the US probably used more drugs in livestock production than many parts of the world. And so for them now to be leading this drive to remove antibiotics is quite revolutionary. And it's having a big impact. Now, probably about half of the chickens raised in the US every year, half of the farms that raise them, will use a vaccine at least once a year. And that's going up year on year. You have chains like Kentucky Fried Chicken, which are making the commitment that the chicken they use will be reared without the use of medically important antibiotics. Of course, quite what that means, I don't know. It's fairly vague. But it sounds good on an advert. But it's clearly the way things are going. Of course, the point is that ionophores, which are used to control coccidia, are considered antibiotics in the US. So there's a big, big change coming. That said, we're not seeing that change quite yet here in Europe. The difference being that we don't have those same wild-type vaccines licensed here in Europe. We only have attenuated vaccines. Now, of course, they are actually much safer. They've been attenuated in most cases by selection for the ability to replicate quicker than normal wild-type parasites. So they're termed precocious. And the way in which they do this is by having one less round of asexual replication. That means they produce less parasites, they produce less damage, they don't cause disease, but they do still go through that life cycle I showed you, and they will still be able to immunize and protect the chicken. But there's some problems. By attenuating these parasites, it makes it even harder to actually produce them in the chicken system that we use. So they're actually relatively more expensive, and it's relatively harder to produce these vaccines. I think that's why, as we talk later on, there's interest in developing new vaccines for Eimeria. But for the time being, it's very popular. It's something of a growth business. Now, the first vaccine registered in the UK was Paracox. There's actually a series of different formulations of Paracox available. But Livacox, that's been around for a long time for the Czech Republic. From Spain, we have Hiprocox. I'm never quite sure what this advert's trying to portray here. I'm not sure if this is the happy-go-lucky vaccinated chicken, and this is the one that's not vaccinated or what, but it's certainly an interesting image. And we've also got Hoover Guard, which is probably the newest kid on the block, and they're actually Australian parasites that have been brought across, reformulated, and are being used in European, but actually they're registered here in the UK markets as well. Now, there's a lot of detail about Eimeria. That's not the only live parasite vaccine out there. There's several others. So there's examples like dosing animals with perhaps a virulent parasite and then treating. So there's things like Theileria, so the Maguga cocktail, for example, which is 3 lines of Theileria, or 3 populations of Theileria, which are combined, used to infect animals, and they're then treated. And that's to give them some level of immune protection. There's naturally attenuated parasites. So Toxovax is an example here. This is a strain of Toxoplasma that was cultured many times in vitro. And through that process, it lost the ability to convert from the acute tachyzoite phase to the chronic bradyzoite phase. And it made it a really good natural vaccine candidate, in that it doesn't persist in the animals it's used in. And you can see at the minute it's primarily used in sheep. There's also a radiated parasite vaccine. So this is Huskvac, that's a Dictyocallis vaccine. This contains L3 larvae. So we're branching to nematodes now, getting out of my natural comfort zone. But again, the point here is they've inhibited the animal, so the parasite, so it shouldn't cause disease, but it's still able to induce that immune response. And I think that's the important point about all of these live parasite vaccines, in that by going through all or part of their natural life cycle, they're able to induce a robust immune response. But the key point is we're required to produce the parasites. Now, as I said a few minutes ago, some of them grow in cell culture, like the Toxoplasma vaccine. Many of them don't. And so it requires us to use animals to produce the vaccine to protect animals. So what other options are there? Well, there are some subunit protein vaccines out there. These examples are using native proteins. So they're proteins that are actually produced by the parasite. So the first example is Leishmune. This is a Leishmania donovani vaccine. You can purify a fucose mannose ligand from the parasite when it's cultured and use that in a vaccine for dogs. The second is Canaleish from Verback, and this is perhaps a similar theory, but it's a different parasite. This is Leishmania infantum, and you can purify the total excreted and secreted proteins from these parasites. And again, you can use them to vaccinate dogs. This second one's available in the UK, but it is important to note you can only use it in dogs that are not infected. So it's all for use perhaps if you're taking dogs on holiday under the pet passport scheme, things like that. Getting a bit closer now to more done. I want to be careful not to say more door, but more done. We've got obviously Barbavax. This is focused on Haemonchus contortus. This is proteins that are purified from the intestinal walls of the worms, and they're used when they vaccinate the animal to induce high levels of circulating antibodies. And these antibodies are then ingested by the parasite when it takes a blood meal. It binds to the gut of the parasite. And while it doesn't actually necessarily kill them immediately, what it does is it interferes with digestion, uptake of nutrients, and it essentially will sort of starve the parasite and has its major impact on reproduction. So it's a sort of population level vaccine, if you like. If you can reduce the reproduction of the parasite, then you actually overall reduce the burden. And this sort of vaccine is called a hidden gut antigen. And that's quite important, because if it's a protein that's only inside the parasite, then it's something that's not normally seen by the host. What that means is you don't get a natural boost to the immune response to this vaccine, so you need to give regular vaccinations to top up the level of immunity, because it is all about maintaining really high levels of circulating antibodies. That's not the only hidden gut antigen. There's another one. This is actually a recombinant antigen. So this is a product called TickGuard Plus, and it contains a recombinant protein from Rhizocephalus microplus. It's called BM because it used to be called Buophilus microplus. And again, this is a gut antigen from the tick. And in exactly the same way, if you have high circulating antibody levels, when the tick takes that blood meal, it ingests antibody that will bind in the gut. And again, it interferes with digestion, nutrient uptake. And overall, you'll have a reduction in parasite replication. But the same problem, you require multiple immunizations with this product to keep that circulating antibody level high. Well, I think it's fair to say, while you do need these multiple immunizations, it's quite ambitious to be able to vaccinate against something that doesn't actually live within its host and just hops on occasion to take a blood meal. So it's a challenging system. One other veterinary parasite vaccine is Sysvax. This is focused on Taenia Solium. In this case, it's, again, a recombinant protein, but it's targeting an oncosphere antigen. So this is when the parasite first gets into its intermediate host. It's called T-Sol18, so it's Taenia Solium. And the idea here is this is used to vaccinate pigs, so that you'll protect pigs from being infected. And I guess we have two levels of interest here. Obviously, we don't want the pigs to get infected, but this is a zoonotic disease, and a large part of our interest here is actually reducing the risk of human infection, so human exposure to Taenia Solium. So that's a brief canter through veterinary vaccines as we stand now. I think, as I said before, there's not a vast number of them, but there's a developing range, and it seems optimistic that there could be more in the future. But what about the future? Well, we're obviously here at a Veterinary Vaccines Network. It's in partnership with SAPHIR, and SAPHIR It's focusing on a mix of viruses, bacteria, and parasites to develop vaccines for. We'll talk more about that in a minute. There's also PARAGONE, that's more focused on parasites with a range of nematodes and arthropod parasites. But I'm going to talk specifically about the work we've been doing in SAPHIR. And in this case, we're focused on chickens. Now, with a background to poultry production in general. We rear poultry in many different ways, from really high stocking densities in very commercial settings, from broilers, or over here perhaps laying chickens, through more extensive systems, sort of barn systems, free range, organic, and through to perhaps those chickens that people keep at the bottom of their garden, picking around. There's a really broad range in the life that chickens would have in this country, around the world, and also it's going to impact fairly obviously on the sort of pathogens that they're exposed to. Now, we've already gone through the quiz bit about how many chickens there are. So there's probably more than 62 billion chickens produced in the world every year. And if you look at the groupings here, we have lots of broilers. Broilers, that's chickens raised for meat, are the most numerous of these birds. We have layers, there's a small number of breeding stock, and there's other birds, sort of pet birds, fancy chickens, things like that. But certainly for these top three, the way in which they're produced is related to what's quite a familiar breeding pyramid. And here you have a very small number of chickens at the top of this pyramid, and they're maintained by a very small number of companies around the world. There's very high levels of selection on these birds for production traits, for health traits now, perhaps resistance to pathogens. These are then cascade down as they have various crosses between these pedigree lines through great-grandparents, grandparents, parents, or the traditional breeder birds, till you actually have the producing animals, the laying chickens, the broiler chickens. So these are mostly hybrids at this point. But what's relevant throughout this pyramid is, of course, pathogen control. Now, at the top of the pyramid, these birds are perhaps kept under much higher levels of biosecurity, so you wouldn't be expecting there to be a particularly high pathogen challenge, but it'll be there nonetheless. But as you come down and as the numbers increase and the intensity increases, then so too does that challenge by pathogens, so we need more control. And of course, if you're a chicken, there are already a lot of vaccines available. So this is a table taken from the Merck website. It's actually the US branch of Merck. I realize it's hard to see the detail, but I'm using it to emphasize a point. And that is that we already vaccinate chickens very intensively. A broiler breeder, that's what's halfway up that pyramid I showed you, is already vaccinated against at least 11 different pathogens, in some cases more. Layers are routinely vaccinated against at least 9 pathogens, again, sometimes more. Broilers, at least 4. And of course, we're a bit greedy, aren't we? We want to vaccinate against additional pathogens. Campylobacter, avian influenza. We want to have vaccines to protect against these. And we're particularly interested in those that are zoonoses. So Campylobacter is a good example. We now recognise that it can be pathogenic in chickens, but that's not been our primary concern. Our concern for Campylobacter, in many cases, of course, what it can do to us if we ingest it. So we have a system where there's currently lots of vaccines, but we want considerably more. So we want to improve current vaccines. We want to develop new vaccines. And of course, this is now being subjected to extra pressure by this drive to replace antibiotics. And the particular example we're focusing on here is Eimeria. So some dates and some facts and figures. In 2008, there was a report in the EU about whether or not anticoccidial drugs should be removed from use in animal production. Now, it took a long time for this to work its way through, and in the end, the decision was not to ban anticoccidials. So in December 2012, that verdict came through, and I think to a large extent, this was informed by what had happened with Histomonas, another parasite, where Histomonas had been under fairly good control. We removed the drugs that held it in check, and actually a parasite that we'd nearly forgotten about has become a big problem again. And so saved by our knowledge of Histomonas, these anticoccidials, or the ones that are still in use, have survived for the time being. But as I said, if we look at other parts of the world, these changes are coming in. So the US, the FDA introduced its first ban on an agricultural drug in 2005. We now, not from legislation, but driven by producers and consumers, there's this no antibiotics ever drive, where there's a big appetite now for, perhaps unfortunate phrase, but there's a big call, a big demand for livestock products that are reared without antimicrobials at any stage. And that's important because the drugs that we use to control Eimeria, certainly in the US, are considered to be antibiotics. And so you do have this drive, say, the Kentucky Fried Chicken example. Again, back here in the EU, in 2016, another consultation was launched, and this time to look at the guidelines of how in which we use anticoccidial drugs and whether that's going to change. So this could be to do with the breadth of the portfolio that we use, how we use them, residues, things like that. So if we're going to develop vaccines for parasites, of poultry in particular, but also livestock, what do we need? Everyone can wake up again. If I've got a vaccine, say, right, I'm going to produce a vaccine for poultry to control parasites, what does it need to do? Apart from make me lots of money, obviously.
Provide protection?
Protection, yes, it's fairly obvious, isn't it? It has to be effective. Anything else? Safety important, yep. Cheap, yep, yep, you're on the nose now, yep, cheap's important.
Easy to administer?
Easy to administer. I won't prolong the pain anymore. So yes, it has to be effective. Absolutely. No, there's no point with a vaccine that's not effective. No one will buy it. When you move to livestock, and particularly to poultry, there's an additional consideration, and that it has to be able to be given easily. A lot of the experimental vaccines we develop, you can get good protection in a chicken if you inject it 4 times with a recombinant protein. But that's never going to happen in the real world. And it has to be something given very simply. So one example of this is a vectored vaccine. It could be a live vectored vaccine, it could be in the diet. But if we look at vectored vaccines, there's some other important points. Now, we want it to be host-specific, ideally. We don't want our vectored vaccine to perhaps be replicating in other non-target hosts. Us, for example. We want that vector vaccine to be immunogenic. You know, if the vector itself has adjuvanting properties, that would be ideal. We want it to be non-pathogenic. And fairly obviously, we don't want to have any harm caused by the vaccine we're using. And ideally, it should be self-limiting. And perhaps overall, we want our vector, in this case, to be able to be made multivalent, so we can actually deliver multiple vaccines or multiple antigens at the same time. Now, there's already viral vectored vaccines out there, certainly being used quite widely, led in the poultry industry. But there are some issues here about the capacity for multivalence. Now, the antigen size and number you can express are very low. So in many cases, you might need multiple viral lines. There are options for bacterial vectors, so Salmonella is used as an experimental vector, for example, but you can have issues about whether it's self-limiting. So what I'm going to talk about is how we're taking Eimeria. So I've already spoken to you about the current live vaccines of Eimeria and how we can take those current live vaccines and actually engineer them to do a bit more. So that if you use a transgenic Eimeria vaccine, you can actually deliver antigens that are perhaps protective against other pathogens. So we just step back for a minute and consider, well, where are we technologically with Eimeria? Can we create these transgenic lines? So for a closely related parasite, Toxoplasma gondii, the tools to create transgenic parasites have been around for a long time, more than 20 years. They're very well established, and they've been a very valuable laboratory tool. In fact, there was actually a patent submitted in the 90s about possibly using transgenic Toxoplasma to deliver vaccines. Nothing's come of it as yet anyway. I think the notion of being injected as a human with Toxoplasma is probably going to be quite a PR challenge. But it's certainly technically possible. But for Eimeria, we lagged behind for a long time. And this is primarily because there was an obligate in vivo phase in the parasite's life cycle. So that same limitation that I described when we produced the live parasite vaccines is the case here. It has to go through, in this case, a chicken. That said, we have developed stable transfection strategies for Eimeria. This is currently based on random integration. We don't yet have ways of targeting the insertion into Eimeria parasites, but you can produce stable transgenic parasite populations. We have some examples down here with different coloured fluorescent reporters. Now, just to take a simplistic approach, for those of you who are not so familiar with transfection, we start off with a construct. So this is normally perhaps a plasmid, a bit of DNA, and it will include our transgene. So it could be an epitope, a string of epitopes, could be a fraction of a protein coding sequence or a whole protein coding sequence. We then need the sequences that flank genes that we use to express these proteins. So we start with a 5' flanking sequence, which will include a promoter. So that determines which part of the parasite's life cycle it expresses in, and a start codon. And you also have a 3' flanking sequence. This might contain a stop codon and perhaps a polyadenylation signal. So we're looking at stability of the RNAs. And in many cases, we have a selectable marker, or several selectable markers. It could be fluorescent proteins, it could be drug resistance. There are ways in which we can enrich parasites and we make them transgenic. So that's our construct. We then have the cell. Unfortunately, we can't transfect oocysts or sporocysts within them. The thick walls preclude us from doing that. So we have to hatch sporozoites, which is the first motile stage in the life cycle. So if we consider a sporozoite as just a traditional eukaryotic cell, we've got a nucleus, we've got a cell wall. We can then use electroporation to drive this plasmid construct into the cell, and with a bit of luck and a bit of know-how, you can produce a transgenic cell. Now, this has actually been quite a slow process. The first transient transfections of Eimeriia was reported 20 years ago, in 1998. It then took 10 years until we had stable transgenic parasites. And on that timeline, 2018 should be a really big year. And happily it is. So Ivan, who's done a lot of the work we're doing there, he's sitting looking slightly embarrassed at the back of the room, has actually had a paper this year, which I think could be the landmark 10-year increment. The challenge will be to have another one before 2028. So the slides I have now are actually stolen from Ivan. I've got a bit of background first. We didn't start off with Eimeriia as a vector for anticoccidial vaccine. We started off using it as a vector for other pathogens. So the first model we used, we actually took a Campylobacter antigen. So this is Campylobacter jejuni antigen A. This is a well-recognized vaccine candidate for Campylobacter. We put it and transfected it into Eimeriia tenella. We then used that transgenic parasite to vaccinate chickens. When we challenged them with Campylobacter, you see a 90% reduction in colonization of the Campi. So I mean, there's still a really high level of Campylobacter colonization. In itself, it's certainly not the answer, but it does show proof of principle that we can deliver an antigen and stimulate an effective immune response. We moved on. This is work by Virginia Marigan-Hernandez, and she was looking to take different proteins. Again, these are recognized vaccine antigens that are already used in commercial vaccines against infectious bursal disease or infectious laryngotracheitis. And in this case, the secret is a bit like those hidden gut antigens I mentioned earlier on. The aim is to stimulate high levels of circulating antibody. And in this system, we found we could stimulate antibody that would recognize the recombinant proteins, which in some ways was a success. But we weren't able to get to the point where we actually tried a challenge experiment because the circulating antibody levels were never really high enough. So we had some issues in that we had some inconclusive immune recognition. You know, we don't know precisely how the vaccine's working, how it's been recognized. And in certainly the second example, we had quite low antibody titers. And one idea for this was that actually the promoters we were using to drive this transfection weren't the best you could have had. They were quite low levels of expression. So we were interested in improving this vaccine vector, and that was the work that was focused on in SAPHIR. But of course, that's only part of the story. What antigens are we going to put in? So our interest was actually to try and improve anticoccidial vaccines. And so we could take proteins that protect against some Eimeriia species and put them into a different species, so you could produce one parasite line that will vaccinate against multiples. And we want to know which antigens to put in. So there's lots of anticoccidial vaccine candidates in the literature. And to try and improve our chances and give ourselves the best antigens, we wanted to look to see when the protective immune response actually happens in the parasite's life cycle. So to do this, we gave a deliberate, sort of like a vaccinating infection with a low number of Eimeriia maxima parasites. We then challenged these birds a few weeks later with the same parasite. So we're looking at effectively a homologous challenge, and we know this gives a very strong immune response. And the graph I'll show you in a minute, the red line relates to parasite genome number, so parasite replication in unimmunized birds, and the blue line is in immunized birds. And you can see straight away, when we look at our first measure, so this is hours post-infection, starting off when a bird's first infected at time 0. Our first time point was 2 hours later. And there's already a lot of parasite genomes in the cells. So this is within a section of the gut where we expect the parasite to be replicating. And then if we look and take 2 hourly counts, for the next 18 hours or so, it doesn't really change. So it's clear the parasite's got down, it's got in, perhaps it's starting making itself at home, but it's not actually replicating yet. And it's only around 20 hours we start to see a difference between those unimmunized birds and the immunized birds. Now at this point, this is our first point of interest, so we'll be interested in proteins that are perhaps expressed and stimulate immune responses that are relevant to this part of the life cycle. But you have to consider that a parasite that's not replicating here might actually have died anywhere during this window. So our first priority were antigens that are expressed very early in the parasite's life cycle. You then actually see as we carry on, the parasite genome number dips again here, so there's some evidence from the life cycle that the parasites actually leave the gut and migrate around the body for a little while before returning to the gut. And you can see now that as parasite genome numbers are eroded in the gut itself in the immunized birds, actually it starts to take off in those unimmunized birds. So it's clear that you'd be wanting to find parasite antigens that again are important later on in the life cycle. Perhaps now ideally in this window. So our priority was to find vaccine candidates that are important that are expressed at these two parts of the life cycle. I actually have quite a large panel of antigens. I'm only going to mention two of them. The first is apical membrane antigen 1, or AMA1. Those of you who perhaps know anything about malaria vaccines will know AMA1 is a very well-known vaccine candidate. It hasn't actually worked out that well for Plasmodium because it's very polymorphic. But we found in some other studies that actually it's not very polymorphic in Eimeria, so it is one of our favourite candidates. And we can see if we use it as a recombinant protein for Eimeria maxima, we get a 42% reduction in parasite replication. I'm comparing here immunised versus mock-immunised chickens. And if we take the Eimeria tenella ortholog, so different AMAs, you take the Eimeria tenella AMA1, you can see a 66% knockdown in parasite replication. Now AMA1 is expressed in sporozoites. It's only expressed in that first stage of the life cycle. So that hits one of our first windows, but of course it doesn't cover the rest of the life cycle. The second protein we mentioned is immune map protein 1. As you can guess from the name, we don't know what it does, but we do know that if you vaccinate with it, you can stimulate robust immune protection. And actually at a level quite similar to AMA1. So in Eimeria maxima, we see about 45% knockdown in parasite replication. In Eimeria tenella, about 67%. And actually this is an average. There's been several experiments, and it ranges from the mid-50s to the mid-70s in the level of protection you can get. And I say there are several other antigens we're working on, but I won't bore you with those now. So we know what antigens we're interested in. But a key job was how we were going to refine and improve this construct for expressing proteins in transgenic Eimeria. So to cut a long story very short, we essentially have two parts in this construct. There's one where we use a fluorescent reporter, this is an M-citrin, and this is under control of micronein protein 1 promoter, so that's a high-level promoter. And this is a transfection control. It hasn't got anything fancy about it, but it shows if a parasite is successfully transfected, that it will then be able to, you can sort it, you can see it's transgenic. The second includes a micronein protein 2 promoter. And this is the bit that's new. Again, it's a high-level promoter. And this is attached to an M-cherry reporter. Now, we then have either on its own, which would be expressed in the cytosol of the parasite, or with a signal peptide that would lead it to be secreted from the parasite, or with signal peptide and a GPI anchor. So it should be secreted and then expressed on the surface of the parasite. And the idea of these is that we can perhaps control the way in which the protein is exposed to the host's immune system, and perhaps try and give it a greater chance of stimulating a response. And then Ivan took, in this example, AMA1, and he actually inserted it between, or just before the M-cherry. And after a lot of work, a lot of time on the microscope, these are pictures that Ivan has produced, and you can see this top parasite here, this is AMA1, it's fused to the M-cherry, so it's the M-cherry you can see. And that is a largely cytosolic distribution, and you can see that's what emerges in many cases with the citrin control. Not 100%, but a fairly good merge. We then have where it's secreted. In this case, it's within the oocyst, so it's being secreted within the sporocysts but staying inside the parasite. And then we have it secreted and actually attached to the surface of the sporozoids. So we had a series of 3 different constructs. Now, to give you an example of how we tested these parasites, we then put this into a vaccination study. So they was using white leghorn as 15i chickens. They're actually produced on this campus at the National Avian Research Facility. We chose them because they're very susceptible to Eimeria maxima. We then had a range of different studies. So there's the 3 different constructs of AMA1. We had an empty vector as a control, and then had a group that received PBS only and will not be challenged, and PBS only that will be challenged. And we then vaccinated these birds on several occasions. This is to mimic the recycling of the parasite that would happen in the field, because of course these are in cages, so it wouldn't naturally recycle. We then gave a fairly low parasite challenge. In this case, it's 300 oocysts. And the point being here, we wanted to look at parasite replication rather than pathology. Now, we mentioned earlier on about vaccines, they need to be safe. So in this example, we can see here that when we vaccinated our animals and look at weight gain during the vaccination process, there was no significant difference between any of these groups. So our 3 test vaccines, the empty vector, and the unvaccinated groups. So based on this criteria, we can say the vaccine was seemingly safe. Obviously, that's not a commercial level safety trial, but it's a good start. What we were interested in was this level of protection. So we're now looking at oocyst outputs. This is the number of parasites produced by the chickens. And you can see from unvaccinated, so birds who just received PBS, there was a considerable reduction in all 3 of our vaccinated populations. And actually, even the empty vector did have some knockdown. So there is some adjuvanting effect here, potentially. There is some cross-protection between the Eimeria tenella and the others. But when you add that maxima antigen, we then saw a significant reduction. The bad news was that actually it didn't seem to matter which of our constructs we used. They all gave good protection. And you can see at the level of protection, this percentage is very similar or slightly better than we've achieved using this antigen with recombinant protein vaccines. And this trial has been done several times. It was actually done originally with slightly different, simpler constructs. And I won't go into too much detail, but again, you can see there was a significant knockdown when, in parasite replication, when you have the antigen expressed versus either the empty vector or unvaccinated chickens. So on the face of it, we're very happy. There was one antigen, we can give good protection, because we want to know how it works as well. And you can look here now, we're looking at Western blots, and this is either against the pure recombinant AMA1 protein or against whole sporozoite antigen extracts. And you can actually see there's a very mixed response here. Yep, so actually we don't see very strong antibody responses stimulated by these vectored vaccines against the particular antigen. And that fits with what we know from that virus work that was done earlier on. And of course that's quite important. It means we can't rely on this vector system to generate high levels of circulating antibodies. But we do see that protection, so it's much better for perhaps T-cell responses. We look at levels of serum interferon gamma, and this is a bit late in the study. It's quite complex, but I think the message we'll take home from here is that actually all of the interferon gamma you can see in birds that have been vaccinated several times have very low levels of interferon, and that's what you would expect. So in a primary infection, you have a high level of interferon gamma. On subsequent infections, you don't really see much interferon being produced. So that's pretty much the same for all. What is interesting is after the challenge, because that's the first time they've seen Eimeria maxima. You know, all the vaccinations were Tenella. And in this case, again, you don't have a high level of interferon. So it does suggest we're stimulating perhaps something more akin to a natural type immune response, despite the fact we're expressing the maxima antigen in Eimeria tenella. And this is very much a focus of future work as we move on. Just a word again, so some work that's been done in SAPHIR is looking at serum interleukin 10 as well. We can see that it's low after the final vaccination, but then it does increase in each of those challenge situations. But the key point to take from this is that it seems to follow that pattern if it's with a transgenic empty vector or with the AMA vector vaccine as well. So it appears to be down to exposure to the Tenella vector, not to the antigen itself. Now, if you're interested in this work, Ivan's just recently published it. He was also did well. He got the front cover of IJP with this quite striking picture. So we've now got some quite brightly coloured Eimeria appearing on the journal in the next issue. I'm sure Ivan can send you a PDF if you're interested. So to summarise, development of anti-parasite vaccines is very challenging. Yep, that's why many of us are here. That's why we're not currently lounging on our yachts in Bermuda or wherever. I hear that? Just finishing, you all right? There's many targets for parasites. Many parasites we're interested in. Very few vaccines available at present. And as we said, those vaccines need to be cheap, they need to be safe, they need to be easy to deliver. We think vaccine can act as a very effective vector, but maybe not if we're looking for specific humoral responses. So who knows, maybe one day we'll go from Paracox 8 to Paracox 1, possibly. Just as an aside, so I lulled you there was one more slide. There's 7 Eimeria species that infect chickens, and there's always a question, how many do we need to vaccinate against? Yep, so we can see one antigen, AMA1, can give us between 40 and perhaps 70% immune protection against one species. Do we need multiple antigens for multiple parasites? They could get quite complex. But to put it in context, the drugs that we use to control Eimeria really give, again, an imperfect control, between 53 and 98% protection, even in a naive parasite. So while we're not in the same league yet, it's quite close. And as sort of good news, if we've got incomplete immune protection, then hopefully it will also prolong the lifespan of those vaccines, because we won't be automatically selecting for vaccine escapees. And as I say, we're greedy. We might be wanting to include vaccine antigens for viruses, for bacteria, and even for arthropods. So just to finish up, there's information about this work on the SAPHIR website. There's a lot of work done on a wide range of other parasites through PARAGONE. And again, you can find information about that on the PARAGONE website. And just to acknowledge that all of the work I've shown in the latter half of this presentation has been done by Ivan. Some of the immunological work has been done here at Roslin by Sung-Won. We've actually been lucky enough to tempt Sung-Won down to RVC, so he's working with us there now. But there's been quite a large group of people who contributed to this work, primarily funded by Horizon 2020, but there's a lot of sort of background funding, if you like, from earlier projects. So I'd like to thank you for your attention, and I'm happy to answer your questions. I'm really disappointed we're not using this. Apparently you can put the microphone in that and throw it around the room. That would have been exciting. See who's awake as well.
Put it on my head.
Well, I would rather, I prefer the small one. But so my question is related to some discussion I heard about the breed selection of birds and a wish to select more and more slow breed birds, especially in the US. Whether it is in relation to also drug use, but also to more welfare kind of issues. Could you comment on the possible interference this would have either on the utility or the use efficacy of the vaccine?
So it's a very interesting topic, and it's something we're working on in other studies. So we're working, again, with partners at Roslin to try to map resistance traits in chickens, in modern chickens, to Eimeria parasites. And what started off as a fairly simplistic study has actually become quite complex. I'm not sure if Andrew's here, but every time we meet at SAPHIR meetings, we seem to go around in circles and it gets more complex. Because there's several phenotypes of interest when we talk about resistance to a parasite, like Eimeria. Is it resistance to the parasite replicating? And that's really important. That's what we're talking about there, because that, if a parasite replicates at a high level, it contaminates the environment, it spreads to others, and they all get sick. But that last bit, get sick, is the key, because actually a farmer isn't that interested in how a parasite replicates. They're interested in the economics and perhaps now the welfare. So they're interested in, does a chicken have an impact on its body weight gain, on its food conversion ratio, lesion scores? That's what counts. And it is quite interesting if you look at different chicken lines, I won't say which chicken lines, but you look at some of the major fast-growing commercial broilers, they actually respond very differently to Eimeria, which is surprising because genetically they're not that different. And I think the assumption is that they've been selected for performance on farms in the face of parasite challenge. So while we've been selecting those that grow as fast as they can, we've been selecting that grow as fast as they can in the presence of the parasite. So we've got birds now, it's quite clear, that are able to thrive under a parasite challenge, even if they've got quite high levels of pathology. So, it surprised us considerably. You can have a parasite that receives a high dose, and if you were to lesion score, it would have a lesion score at the very top of the scale, but it's still growing, still thriving. Now, that's a bit of an ethical dilemma, isn't it? Because actually, that chicken is heavily parasitized, it has a lot of pathology, but it's still performing. So there is a challenge there. But when you move to slower growing breeds, I mean, the parasites evolved to live with chickens, and I think in many ways, as long as you can control the exposure of that chicken to the parasite, that'll be a far better concept. Because you do see, if a chicken's exposed to Eimeria, it may have some brief impact on its productivity, but you see compensatory growth afterwards.
But could the bird be resilient without any use of vaccine?
Well, we're getting to the point, but it seems there's always like an arms race. Because if it's resilient, it will then produce more parasites. And there's more parasites in the environment, and those resilient birds will then be exposed to a higher level.
Yeah, could you comment on the prospects for a Cryptosporidium vaccine for cattle and zoonosis? This has been ongoing for a long time.
It fits into the same path. I think there are some promising vaccine candidates, but in many ways we're still trying to understand the complexity of Cryptosporidium itself. We've gone from the point where there was a single Cryptosporidium species to 2 genotypes. We now have an explosion of more than 40 Cryptosporidium species. It may be they're not all true species, and we'll back off from that, but I think where we have antigens that experimentally can give good protection, what we see in the field is quite a diverse challenge, and we need to understand the challenge better before we can come up with the best vaccines. But there is, again, a lot of work gone to that. I know there's an application gone to the European Union to try and explore just that area. You're making people work hard, all questions at the back. If the next person's at the front, then we could really sit.
Taenia, I was very intrigued by the fact that you said we didn't proceed with the challenges when you expressed viral antigens in your Eimeria. Because with these viral infections, it's often the T-cell responses
That's interesting. I mean, so
that are stronger. So I would still challenge them.
So we're working with a commercial partner of this, Merck, and to their mind, their diagnostics for vaccine success are all about antibody.
Of course, because they can't do the T-cell responses. Just because they can't have the they don't have the technology.
I mean, it wasn't our choice. Essentially, they looked at the test, they saw there wasn't antibody, and the plug was pulled.
Okay, we can do it here then.
Well, we still have the parasites. Yep.
Taenia, you have such a vast experience with all these vaccine trials and challenge trials. Could you comment potentially on how diverse, how uniform is the response of the animals to these vaccines? Is there a very uniform response, or do you think there are some animals or some species or some breeds that may respond better to one stage of the parasite life cycle, a vaccine against that, and another one? Is that an issue or not?
I think it is, and I think it's something we're increasingly recognising as an issue. I mean, if you go back to some of the older studies done, again, with the inbred lines here, not only do you see different levels of parasite susceptibility in different inbred chicken lines, but you also see different levels of immune protection against a homologous challenge. But where it gets really interesting is you can see completely different levels of protection against heterologous challenge. So there's some chickens here where I could vaccinate with one strain of Eimeria maxima and challenge it with a different strain, and it's 100% cross-protected. If you go to a different chicken line, it's 0% cross-protected. So, I mean, those are extremes. They're inbred chicken lines. But clearly there are fundamental differences. Now, part of the GWAS study we're doing is not just looking at susceptibility or resistance, I use those words tentatively, in primary infection, but it's also looking at the outcome of secondary infection. That was where there was a cross made between those two chicken lines, the one that had the 100% cross-protection and the 0, and to try and map it. And it looks as if, yes, there are QTLs that will associate with that, and they're completely different to those that associate with the outcome of primary infection. So I think it will be important. It may be it's possible to select chickens for their response to anticoccidial vaccination. Of course, whether that's a good thing, I guess we'll wait and see.
Thanks.
Madeline's coming.
Congratulations with your very nice results, but I don't follow completely. Because you started your talk with saying, Emeria, sporozoites are difficult to produce
Yes, they are.
commercially.
Yep.
And so you target now Emeria tenella,
Yep.
sporozoites, as a vaccine delivery
Yep.
system. But you said it's not the ideal system. But you want to mimic probably the cycle,
Yep.
and therefore you incorporate these other antigens. Now, my first question is it better than the paracox?
So, in answer to your first question, paracox now, depends which paracox you're looking at. The one that's in mainstream
Containing
use.
maxima and tenella.
So the mainstream paracox has 8 parasite lines in it.
Yes.
One of which represents half the cost of production. So if you could take parasite antigens from one species and take that into another one, say paracox 7, at a stroke you would halve the price of the vaccine and the production capacity. If you could actually take all of those and go from paracox 8 with all those lines of parasites to one transgenic line.
Yeah.
Now, you're right, Taenia wouldn't be your ideal choice. You'd go for something that's more fecund, like Amira cervulina, but this is experimental. But at this stage, if you could take one parasite line that protected against all of those, then you've got a model that you can actually scale up.
Okay. Now, the reduction from 2 times 10 to the 7th to 1 times 10 to the 7th.
Yep.
Okay, you saw that, but is that important in the cycle? Because that's only a 50% reduction, and we are still at 1.10 to the 7
Yep.
multiplication.
So it can often be the difference between being infected and becoming immune, or becoming infected and becoming sick. But I think we'd also say this is the start. You know, we're looking to make these vaccines where you'd have multiple antigens.
Mm-hmm.
So the example, the second one was immune MAP protein 1. There's several others that we're looking to put in. It is a good question, and it's something we often debate as to how many antigens will you require. And so the next study the study after the next one that Ivan will be doing is starting to look at that, where we're actually taking vaccine lines that express different single antigens, use those separately and combined, and to see whether we can get an additive, or if we dream about it, maybe even a synergistic effect.
Okay. And then when you were looking at the cytokines, interferon gamma, you were looking in the serum.
Yep.
And you didn't see, but is it in the serum you have to look at?
You would not, so I mean, this is just work that's done at the very end. The next experiment is done, which have real depth. But the point is, in a normal Emeria infection, you would see very high levels of interferon gamma in the serum.
Yeah, but perhaps it's a side effect.
We don't know for sure.
In
but
the serum,
certainly-
I mean in the serum.
But certainly in mice, it's crucial, because if you have interferon gamma knockout mice, now we don't have an interferon gamma knockout chicken, perhaps we're in the right place to request one. Maybe next week after, I don't know.
Okay, thank
But yes,
you.
so we don't know for sure, but certainly interferon appears to be the primary mechanism of protection in that first round.
Okay.
In terms of the interferon gamma, the comment that interferon gamma we measured in the serum level, and in the previous literature clearly shows that primary infection, interferon gamma in the local and then serum is increased, but following the serial secondary or third infection, the interferon gamma level is sharply decreased, unlike IL-10. That's one thing that we found on the same trend in the previous in the Evans experiment.
So I think the key point is it's something that interests us as to the mechanism of this protection. Are we stimulating what looks like a natural immune response just with one Emeria maxima antigen, or is it an unnatural response but still gives protection? And it's important to know that, because it could then lead us to include different immunostimulatory molecules in this, to whether we can bias or improve the immune response.
How many cycles do they replicate? With the tenella that you use as a vector?
So the minute it's wild type, so there's 3 rounds of asexual replication.
Timur, what does the regulatory sort of landscape look like for this kind of thing? I mean, obviously, this is an industry that's very comfortable with viral vectors and things, but for a GM protozoan, what does that look like?
I suspect that will be our biggest challenge. At the time now, we would certainly not, I think, be able to release a transgenic Eimerian into the environment. But that said, some of the big companies like Merck sponsor this, and they're very confident that in time that will be possible. But I agree, I think that we could well get to the point where we have an effective vaccine that can protect against multiple Eimeria species, and the challenge would be whether it can be registered to be released. I mean, there's certain options. You can sort of talk about suicide genes, things like that can be put in, but that's difficult because, of course, we require the parasite to recycle to get the level of immunity you need. So it doesn't go hand in hand. But I think that will be the biggest challenge.
I just wanted to add a remark from the remark of Andrea on the individual, the variability of the response. And you were saying that there was, from your GWAS results, that the different QTL for primary and secondary response. So in a sense, you tend to say that the genetic control of a response to the vaccine and to the challenge might be different. So to your question, saying would that be possible or even interesting to select birds for a better response to the vaccine, I would go further. We should see if it's possible or interesting to complement the genetic, a better response to the vaccine and to the challenge. Is there a synergical effect? And that is, I think, the key point that we should try to test. But so far, and now we start with SAPHIR, but we have been quite working in different fields, and now we finally can come up and merge results. But I think that's the idea, is to look on the synergy and not individual improving of the response.
I agree. I mean, I think we've not, as a community, necessarily been very good at joined up thinking. And of course, the other challenge is that the genetic selection of these birds is controlled by a very small number of very big companies. And it's only recently, I think, that they've been won over to the importance of resistance to these parasites. They're saying they seem a lot more engaged than they were 10 years ago.
I was asking always myself, the myriazoids, after the first cycle, do they go intraluminally or do they go in the lamina propria and go to the other cells to reinfect? Because they are different diagrams. The ones of Janssen Pharmaceutics, they draw the myriazoids in the tissue and not in the lumen. And that could have an important implication of the immune responses you induce, because if they stay in the lumen, well...
I would say there is a halfway house there, actually, in that in most cases they don't necessarily go out into the lumen because they're not nearly as robust as the sporozoids. But in most cases they do emerge and then invade neighbouring cells.
Yeah.
But of course they don't necessarily decide that. They might invade a cell and decide, I don't like this cell, and they'll leave that one and go to another cell.
Yeah.
So, and that's quite important. I mean, you're right. In the terms of the type of immune response they're stimulating, that's really important. Because every time there's an invasion process, they're actually producing proteins, they're interacting with the host.
Inflammation and all this.
Precisely.
Things.
And that's why we were so keen on moving where we express this antigen, because the first study, if the protein is expressed cytosolically, you require the parasite to die to release the protein.
And did you do histological sections? From when on do you see CD4 cells appearing in the lamina propria or between the epithelial cells? Well, let us say, you don't have to say they are specific for Coccidia, but when do you see them coming off or increasing the CD4 T cells?
So we haven't done it specifically in this work, but it's around day 4 that you start to see them really, of post-infection.
Post-challenge or post-infection?
Post-infection. After that first infection is where you see. And again,
in immunized animals?
That's in primary. In secondary...
In primary, already 4 days afterwards?
That's where you first start to see them, exactly that. And then in challenge, it is probably a little bit earlier, but maybe 2-3 days, but not that much earlier.
They haven't left in the shop. They stay.
Yep. Well, it depends on how long the gap is. But yeah, I mean, we often have this debate about what is a true secondary challenge or what is like an ongoing infection. Of course, unless you have a clear gap.
And do you see the problem of Histomonas, well, which has naturally is invading and goes to the liver, how they go perhaps inside with bacteria carrying with them and all, but do you see a similar immune mechanism operative in Histomonas
We
as
haven't
in
worked-
Coccidia?
We haven't worked with Histomonas, but I think it is a very similar profile, but it tends not to be as dramatic. You see really high levels of cytokines in Eimerian infection. It seems Histomonas isn't quite as blatant. It doesn't stimulate quite such strong or high magnitude responses.
And is that because they are hidden in macrophages and
To some extent, I think yes.
it's rather a bacterial infection of the liver we get and necrosis of the liver
some
because
extent.
they carry the bacteria with them?
Yep.
Okay.
Damon, is there any production cost to your vaccine process? If you take a healthy bird with no infection, no vaccination versus the vaccinated bird, is there any impact on growth rate or feed conversion ratio?
In vaccination of
In
the
the
chicken.
vaccinated bird compared to the, I don't know what you call a healthy bird because you're saying that coccidia is so pervasive, but
So there's two answers to the story, and there's a European answer and an American answer. In Europe, we use attenuated vaccines, and there does appear to be pretty much no impact on food conversion or weight gain. It depends on some studies have suggested there are, but in most cases there's not. Certainly by the end of the study, there's not. In the American system, they use wild-type parasites, and you can see that there is a brief window where food conversion is compromised, and body weight gain can tail off in vaccinated birds. But the trade-off they have there is they keep birds a little bit longer than we do, and actually they get compensatory growth by keeping them that bit longer. And it depends on which company you talk to, but that compensatory growth will often take them to where they would have been without the parasite, in some examples even a little higher. So some debate about future vaccines in the US is, would you lose that compensatory growth? And maybe indirectly that's part of their model.
and you mentioned the welfare impact.
Yep.
So you've got a bird that looks healthy, it's grown okay, but it's full of parasite,
Yep.
basically. Is anybody looking at that kind of welfare aspect in terms of measuring what the welfare impacts actually are? Because that's probably quite an important issue in certain markets, isn't it?
It is. I think they're starting to. Scientifically, there's a guy called Theo Denmers at RVC who's been starting to look at that. His main grant of that was unfortunately not funded, but we have some pilot funding to do that. But of course, there's a lot of scrutiny overall on health for chickens, which isn't specifically related to Eimeria, but is part of that.